I now call to order the Society's 2,484th meeting in what is now the 152nd year since its founding on March 13, 1871. Good evening, everyone. My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members, guests, and friends to tonight's PSW meeting and to the lecture by Ted Schultz on Convergent Evolution of Agriculture. Let's start by thanking our sponsors, including our full-year sponsors, Mike Helton and Helton Associates, and the Policy Studies Organization in cooperation with the American Public University. Our fall 2022-2023 lecture series sponsor, PSW member Tim Thomas. The anonymous sponsor of tonight's lecture. Please thank them all. I'm also very happy to tell you that PSW has been given a donor match gift by a generous sponsor who is also asked to remain unnamed. All contributions from now through January 31st, 2024 will be matched 2 to 1. So for every dollar you give, PSW will receive $3. Your dollar and 2 matching dollars, up to a total of $20,000 in matching contributions. So now is a very good time to support PSW financially by making a donation during the matching period. You will be receiving additional notices on this opportunity in the near future, and let's all thank our generous donor who has asked to remain unnamed. Thank you. I am pleased to announce the following new members. Ron Ong, a Space Force officer currently with DARPA, interested in behavioral economics, research and development, and engineering, who learned of PSW from the PSW meetup group and from attending PSW lectures. And tonight's speaker, Ted Schultz, who learned of PSW some years ago when his colleague Cameron Curry spoke here, and whose interest will be clear in some small part from tonight's lecture. We welcome them to membership. All members are entitled to a signed copy of Volume 1 of the Bulletin of the Philosophical Society of Washington, that is PSW. If you've not received yours, please come see me after the meeting, and I will be glad to provide you with your copy. Robert Thompson, Vice President, will now present the minutes of the 2,483rd meeting and the lecture by Pri Narang on non-equilibrium quantum mechanics, prepared by PSW member Charles Clark. Robert, the stage is yours.
Good evening, everyone. On October 20th, 2023, in the Powell Auditorium of the Cosmos Club in Washington, D.C., and by live stream to the PSW Science YouTube channel, President Larry Milstein called the 2,483rd meeting of the Society to order at 8:06 p.m. Eastern Time. He welcomed attendees, thanked sponsors, and announced new members. Mark Clampin then read minutes of the previous meeting and the lecture by Lori Locascio on the National Institute of Standards and Technology, prepared by Recording Secretary Cameo Lance. President Milstein then introduced the speaker for the evening, Praneha Pri Narang, and her lecture, Driven Out of Equilibrium, Shining New Light on Quantum Matter. Narang's presentation focused on non-equilibrium states of molecules and materials as probed by laser excitation. This mode of excitation enables one to explore specific pathways through the potential or free energy landscape of a material versus non-selective thermal excitation. The central theme of her lecture was her work on the study of photon-matter interactions in cases of very strong coupling. This work led her to identify systems that exhibit photonic quasiparticles. In those cases, a material system that would usually be described in terms of an electronic state with many virtual excitations induced by an intense laser field can instead be viewed as a strongly coupled radiation matter state with a small number of photonic quasiparticle excitations. In the limit of laser intensities, this central idea reduces to the Jaynes-Cummings model of quantum optics, in which states of atoms and states of the radiation field are strongly coupled by resonant light-matter interactions, specifically when the photon energy is near the difference in energies of two quantum states of the atom. A striking aspect of Narang's work is that she has been able to demonstrate strong radiation coupling with elementary excitations of materials such as magnons, polarons, and Cooper pairs. One demonstrated example of this is manipulation of magnetic order in an antiferromagnet through lattice vibrations excited by a laser. In the ground state of an antiferromagnet, electron spins are oppositely paired. Laser-induced shaking of the lattice induces a ferromagnetic moment that oscillates at very high speed. Other recent experiments of this type have demonstrated phonon-driven superconductivity and ferroelectricity, magnetically dressed vibrational excitation, and strain-driven topological symmetry breaking. Narang has found that familiar optomagnetic phenomena like the Faraday and Kerr effects can have phonomagnetic counterparts induced by phonons generated by laser excitation. For example, in inverse phonomagnetic effects, the phonon mode acts as an effective magnetic field on the spins in the material. This phonomagnetic field induces a phonon inverse Faraday effect. The phonomagnetic effect is not due to the presence of an actual magnetic field, but rather an electron spin correlation effect. Nevertheless, it can lead to spin polarization in solids of the same type as that induced by strong magnetic fields. Narang and her colleagues predict that an effective field, equivalent to a magnetic field of over 100 Tesla, can possibly be generated that polarize the spins of 4f electrons in lanthanide materials. Another example of such a hybrid approach is the magnetic infrared resonant Raman effect, made possible by interaction of magnons with Raman-active phonons. The modification of bulk magnetic properties by light has been of great interest, and a recent collaboration of Narang's group with an experimental group in Hamburg, Germany, has shown laser manipulation of the crystal lattice of yttrium titanate can alter its ferromagnetism. Narang discussed examples of condensed matter analogs of some of the constructs of high-energy physics. She has collaborated with experimentalists to demonstrate the existence of Higgs and Goldstone-like phonon modes in lanthanum tritelluride. It's always in the last place you look. She also discussed proposals to realize axion fields in topological insulators. The axion, named by physicist Frank Wilczek after a brand of laundry detergent, you don't make this stuff up, right? Is a hypothetical fundamental particle that has yet to be observed. Its existence would account for an unexplained symmetry that is observed in the standard model of particle physics. Some quasiparticles in certain materials, such as strong topological insulators, seem to be described by axion-like equations, and Narang discussed the prospect of using axion electrodynamics for next-generation devices, and even in searches for real axions as a possible constituent of dark matter. After the lecture, there was a question and answer period. Several questioners asked about implications of the Higgs, Goldstone, and axion work for fundamental physics. Narang said that no certain outcomes can be stated, but it is striking some new constructs of the standard model are appearing in condensed matter systems and training a new generation of physicists. After the question and answer period, President Milstein thanked the speaker, announced upcoming lectures, thanked volunteers, and asked everyone interested in helping out to get in touch with him. He adjourned the meeting at 9:50 p.m. The temperature in Washington, D.C., 68 degrees Fahrenheit. The weather was mostly cloudy. The live audience in the Powell Auditorium was 54, with 19 watching on the live stream. Views of the recording on the PSW Science YouTube channel in the first 2 weeks following the lecture were 765. Respectfully submitted, Robert Thompson and Charles Clark, on behalf of Recording Secretary Camille Lance.
Are there any comments on the minutes or any questions about them or any corrections? Hearing none, I'll entertain a motion to accept the minutes as read. Okay, a second. All members in favor of accepting the minutes? All members opposed? Minutes are accepted as read and will be posted to the website in due course. And we now turn to tonight's lecture by Ted Schultz on the convergent evolution of agriculture in ants and humans. Ted is research entomologist and former chair of the Department of Entomology in the Smithsonian Institution, National Museum of Natural History, and he is adjunct professor of entomology at the University of Maryland in College Park. Ted's research focuses on the evolutionary history of ants using phylogenetic and fossil data. He is an author and co-author on over 120 publications and several books, including The Convergent Evolution of Agriculture and Insects and Humans and The Fringes of Reason, a whole earth catalog of New Age frontiers, unusual beliefs, and eccentric sciences. Prior to becoming a research entomologist, Ted led a peripatetic life, engaging in a wide variety of endeavors, which I will leave for him to describe in the social hour. Among other honors and awards, he is a fellow of the American Association for the Advancement of Science. He earned a BS in biology at UC Berkeley and a PhD in entomology at Cornell University. All questions will be fielded after the lecture during the Q&A session. Ted, the stage is yours.
Thank you, Larry Milstein and the Philosophical Society of Washington for inviting me to give this talk. Let's see. In biology, mutualism is a symbiotic relation between two organisms that benefits both of them. And it can be more than two organisms as well. And you want to compare that to another kind of symbiotic relationship, parasitism, in which one of the participants suffers a loss. So agriculture is a form of mutualistic symbiosis. Now, you might say, well, I think it's a parasitism because we wind up eating our domesticates. And I think you have to consider facts like this. The biomass of extant humans and their mammalian and avian livestock is 17 times greater than that of all the wild mammals and all the wild birds in the world combined. There are similar statistics for our plant domesticates like maize, wheat, rice, soybeans, etc. So if fitness is representation in the next generation, agriculture is a mutualism. Agriculture originated around 23 independent times in humans. This includes the origins of seed crops all over the globe. And here, let me test out the, yeah, there we go. So seed crops all over the globe. It also includes the many origins of vegetatively cultivated crops all over the globe, crops that we propagate through cuttings. Less well appreciated, agriculture originated approximately 20 known times in non-humans, including many times in insects. This includes farming by damselfish, snails, polychaete worms, of algae and fungi, and most relevant to tonight's talk, by fungus-farming termites, fungus-farming beetles, and fungus-farming ants. Tonight, I am going to focus on the fungus-farming ants, partly because they're arguably the most successful of the non-human agriculturalists, but mainly because they're my favorite organisms on the planet. This is an exclusively New World clade of 247 described species, many, many more undescribed species awaiting discovery. All fungus-farming ants are restricted to the New World, and they all obligately depend on the cultivation of fungi for food. This includes the charismatic and well-known leaf-cutting ants. Leaf-cutting ants in the genus Atta can have huge nests with thousands of underground garden chambers, like in these pictures. Here's a schematic diagram showing just how big these nests can get. Here are the thousands of garden chambers. Here are these huge waste dump chambers, and there's a human for scale. These nests of Atta have been called superorganisms. An Atta colony is the equivalent of a large herbivorous mammal like a cow, and in terms of its longevity, it can live 10 to 15 years or more. In terms of its biomass, if you weighed all the ants and the fungi too, it would weigh as much as an adult cow. And in terms of the amount of fresh vegetation that they harvest and take into the colony every day. But the leaf-cutting ants are not directly consuming the leaves that they bring into the colony. Instead, those leaves are being digested by a fungal intermediate. But most fungus-farming ants are not leaf-cutting ants, and these non-leaf-cutting fungus-farming ants are very inconspicuous. They can have very small nest entrances like this. They can have very small colony sizes. Their gardens can consist of a single garden in a single garden chamber beneath the soil. Here are some more examples of garden chambers. I'll talk more about, we spend a lot of time in the field digging up fungus-farming ant nests like this, and I'll talk more about that later. Here's an example of a fungus garden that the ants have placed on the underside of a rotten log. And here's another example of a rotten log nesting fungus-farming ant. But whether you're a leaf-cutting ant or a non-leaf-cutting ant, the suite of behaviors required for cultivating fungus gardens is remarkably complex. The ants have to forage for the appropriate organic substrates. The non-leaf cutting ants forage for things like insect frass, tiny little plant parts that have rained down from the canopy, seeds, things like that. In some cases, the ants have to prepare the substrate. Then they have to incorporate the substrate into the garden. They have to go off and find healthy growing mycelium from elsewhere in the garden, bring it back and plant it on the newly added substrate. They manure the gardens with their fecal droplets, which contain high concentrations of proteolytic enzymes. They have to remove the spent substrate. Once it's been consumed by the fungus, it's a source of disease. They have to harvest the fungus for food and feed it to their larvae. And daughter queens have to transfer fungus from their mother's nests to their nests, and I will talk more about that later. The first person to write about, give a detailed account of the leaf-cutting ants was the great 19th century naturalist Henry Walter Bates, who published The Naturalist on the River Amazons in 1863. And he wrote, The leaves are used to thatch the domes which cover their entrances to their subterranean dwellings, thereby protecting from the deluging rains the young broods in the nests beneath. So he was wrong about that. He thought that they served some kind of a waterproofing function. It remained for another great 19th century naturalist, Thomas Belt, to discover ant agriculture, which he did in 1874. And I would just like to point out that that's 100, I think if I have the math right, it's 149 years ago. Next year is the 150th anniversary of the discovery of fungus farming ants. And I don't know about you, but I intend to celebrate. And in that same year, well, wait, let me tell you what Thomas Belt said. He said, "I believe the real use the ants make of the leaves is as a manure on which grows a minute species of fungus on which they feed, that they are in reality mushroom growers and eaters." In that same year, simultaneously, yet another great 19th century naturalist, Fritz Müller, also discovered ant agriculture. He communicated this discovery to the then very young journal Nature through his correspondent, Charles Darwin, and he wrote, "As to the leaf-cutting ants, I have always held the same view which is proposed by Mr. Belt, namely that they feed upon the fungus growing on the leaves that they carry into the nest." In 1890, Fritz Müller's nephew, Alfred Müller, came over from Germany to visit Uncle Fritz for 3 years while he conducted research on the cultivars of the fungus farming ants. The work that he produced remains a seminal scientific classic. He presented figures of the fungus garden, the fungi that grow in the garden, as including fungi other than the fungal cultivars. He collected, photographed, and described as a new species mushrooms that he found growing out of an abandoned leafcutter nest. He named this fungus Rosides gongylophoros. We have since placed it in the genus Leucoagaricus, so Leucoagaricus gongylophoros. Mushrooms do occasionally occur in laboratory cultures of these fungi. And based on these rare mushrooms, various mycologists over the years have tried to identify the fungi grown by fungus farming ants with varying degrees of success. As has been known since the late 19th century, a daughter queen, when she leaves her mother's nest, carries a bit of her mother's garden with her. She carries this in her infrabuccal pocket. This is a pocket in the mouths of all ants. So this very huge Atta queen, when she left her mother's nest, she carried a little bit of fungus in her mouth. She left on her mating flight. She mated, she dropped to the ground, she shed her wings, she excavated a chamber in the soil, she spit out that bit of garden, and she took care of it, manuring it with her own fecal droplets, also feeding it trophic eggs. These are non-viable eggs that she produces, and she continued to do this until the first brood of workers emerged and took over the task. Ant agriculture has a crop disease, a clade of ascomycete fungal species found only in fungus gardens that have co-diversified with the ants since the origin of fungus farming. The disease is called ascovopsis, and it competes with the ants to consume the fungus garden. The ants, fungus-farming ants, culture on their bodies actinomycete bacteria. These bacteria produce antibiotics that the ants use to control the crop disease Ascovopsis. The ant actinomycetes are closely related to streptomyces, from which humans derive a lot of our antibiotics. The fungus-farming ants have integumental crypts all over their bodies where the actinomycete bacteria are housed, and these crypts are lined with glands that presumably nourish the bacteria. We were able to see these crypts in fossil ants in amber, Dominican amber, which is about 15 to 20 million years old. So in those amber colored figures there. So ant agriculture is minimally a four-part symbiosis consisting of three mutualists, the ants, the fungi that they cultivate, the antibiotic-producing bacteria, and a fourth parasite, the Ascovopsis crop disease. But new consistently occurring microbes are being discovered all the time. Because, understandably, researchers over the years really wanted to know how ant agriculture evolved, for over 100 years, they've attempted to construct ant phylogenies, ant evolutionary trees. Here are two really amazingly prescient such trees from 1912 and from 1963. More recently, there's been a couple of more formalized attempts to come up with ant phylogenies. Here's one from 95 and one from 1998. And with the advent of molecular systematics, there were further attempts. Here's a crude early molecular phylogeny from 1998, and then a better one from 2008. Now, attempts to construct fungal phylogenies have lagged pretty far behind. In fact, it started with molecular phylogenetics. Here's two of the earliest attempts, one from 1995 and one from 1998. Oh, and here's another one from 2012. Now, what we have here is we call a tanglegram. It's two phylogenies juxtaposed, in this case an ant phylogeny and a fungal phylogeny, with little lines drawn in between from the ant to the fungus that it grows. This tanglegram from 2015 includes an ant tree that is based on 4 genes and a fungal tree that's based on only a single gene. And the trouble with all of these early phylogenies is that the available markers could identify small, closely related groups of fungi, like in these colored boxes, but the markers could not tell us how those groups were related to one another. Even so, back in 2010 or even a little earlier, we could see a pattern in which broad phylogenetic groups of ants cultivate broad phylogenetic groups of fungi that are parasitized by broad phylogenetic groups of Ascovopsis. We could also see that although the ants from one group were never found growing fungi from another group, this pattern breaks down within the groups, so that within the groups, closely related ants could be cultivating distantly related fungi, and the same fungal species could be cultivated by distantly related ants. But that only is the case within these groups. We refer to these as agricultural systems, and we would very much like to understand what biological mechanisms enforce the boundaries between them. And you know, these observations of these agricultural systems are based on quite a lot of sampling by quite a lot of people from a number of labs in the United States, in North America, Europe, and South America over the last 30 years. But this tally is from 10 years ago, from 2015. And there's been a lot of work since then, so this is a gross underestimate. Now, even though we don't understand to this day what mechanisms restrict ants from one group from cultivating fungi from another group, we do have some insight into why the pattern breaks down within the groups. And it's because of something called horizontal transfer. Ants are able to, well, based on lab experiments and field observations, ants can occasionally replace their resident fungal cultivars with other cultivars that they obtain from the nests of other ants or from fungi growing ferally in the leaf litter, unassociated with ants. I'll tell you a bit more about that later, but this might happen, say, if a colony's garden dies, or if a daughter queen forgets to bring fungus with her from her mother's nest, or if she brings fungus, but the fungus then dies. So horizontal transfer would violate the pattern we would expect to see if fungi were always faithfully transferred vertically from mother to daughter nest. And if they were always faithfully transferred, we'd expect to see this. Whoops. But that is not what we see within those groups. Well, I'm happy to report that we now have a different, in the last 5+ years, we have a different way of creating phylogenies. We have phylogenomic markers, ultra-conserved elements, or UCEs, that produce, for ants, that produce massive amounts of data from loci sampled, independent loci sampled from across the genome. This allowed us in 2017 to create this phylogeny for the fungus-farming ants, in which virtually every node is maximally well supported and which essentially nails down fungus-farming ant relationships. So our mission since then has been to add species to this phylogeny using an enhanced set of markers. And we now have phylogenomic markers, ultra-conserved elements, UCEs, for basidiomycete fungi. These are still unpublished, but I'm actually planning to submit a paper using them in the next couple of weeks. These markers have allowed us to construct this phylogeny for the fungal order Agaricales. It contains 288 ant-cultivated fungi, and it indicates that all ant-cultivated fungi arise within two families in the Agaricales, in the coral fungi, the Pteriolaceae, and in the Agaricaceae, and particularly in the tribe with Leucocoprinae within the Agaricaceae. The Agaricaceae contain the common grocery store button mushroom that humans widely cultivate and consume. Now here, this tanglegram includes a fungal phylogeny that's based on the one I just showed you, but it's focused just on the ant-cultivated fungi and their close relatives, and an ant phylogeny, all based on ultra-conserved elements. And of course, these lines in between indicating associations between ants and fungi. You may or may not be able to see it, but I've colored branches in red on this fungal phylogeny if they lead to ant-cultivated fungi, and I've colored branches black if they lead to non-ant cultivated fungi. You don't need to see any details here, but I hope you can see this pattern of pronounced fidelity between broad groups of ants and broad groups of fungi, which I previously referred to as these agricultural systems. This tanglegram indicates that the first fungus-farming ant species, this red arrow here, the first species cultivated two different but closely related groups of fungi, the two red arrows over here, and these two separate yellow boxes. For years we've known about these, and we've called them clade 2 and clade 1, but we didn't know that they were cultivated separately and simultaneously. There are 10 genera of ants that practice this lower agriculture in the yellow box, and some of them look like this. They can have relatively small colony sizes of a couple dozen to a couple hundred individuals. They can often just have a single garden chamber like this one, or like this one. We know of 3 cases where free-living mushrooms have been collected that, when DNA sequenced, proved to be the same species as or very closely related to an ant-cultivated fungus. This suggests that although the ants cannot live without the fungi, the lower fungal cultivars are capable, at least temporarily, of living without the ants. This is different from saying that the fungi have not co-evolved with the ants. We just don't know. Many of our domesticated plants and animals are likewise capable of escaping into a feral existence, but nobody would say that they haven't genetically evolved for life with humans. From generalized lower agriculture, we get a number of evolutionary transitions to different, arguably more specialized forms of agriculture, such as this transition to coral fungus agriculture. In the fungus-farming ant genus Apterostigma, some Apterostigma cultivate lower fungal cultivars. Here's an example of one of those species, Apterostigma auriculatum, on its fungus garden. But about 20 million years ago, an ancestor of this group of ants in the genus Apterostigma switched and started to cultivate coral fungi. It took its suite of farming behaviors and applied them to a completely different fungus. The coral fungi, the Pterilaceae, are very distantly related to the Agaricaceae and the Leucocoprinae. And you know, humans do this all the time, transfer background agricultural knowledge to cultivate some new crop, but this is the only time we know that this happened in the fungus-farming ants. And there are actually 2 groups of coral fungi that the ants are cultivating. In one of those groups, Myrmecopterula velohortorum, the gardens have a tent-like protective veil over them that the ants weave together from the hyphae, the fungal hyphae. And that allows these ants to build their nests in strange exposed locations, like on the trunk of a tree here and on the underside of a leaf here. But in this other group, Myrmecopterula nudihortum, there is no veil around the garden. Another evolutionary transition away from lower agriculture occurred in yeast agriculture in these green boxes. And unlike the typical white cottony mycelial garden that you see here, yeast gardens consist of this very different fungal growth form that creates these little yellow nodules, and they consist of disconnected single cells or maybe just a few cells connected to each other, whereas mycelium, which is how most fungi grow, the growth form of most fungi, they're linear branching chains of completely connected cells. Here's another example of one of those yeast gardens. And as in lower agriculture, we know of 5 collections of free-living mushrooms that, when DNA sequenced, fell within the yeast fungus clade, suggesting that in yeast agriculture, the fungi are also capable of living apart from the ants, at least temporarily, and that when they do this, they grow as mycelium rather than as yeasts. They also revert to mycelial growth when we culture them on agar in the lab. And they grow as mycelium on the bodies of the ant larvae in the nests. Now, you could argue that the yeast fungi are not at all adapted for life with ants, that instead they are passively manipulated by the ants, which do something to induce yeast phase growth. But if that's the case, why do the yeast farming ants, a small compact clade of about 20 species, seem only to be able to manipulate species in this correspondingly compact small clade of yeast fungi? I think that this phylogenetic pattern is more consistent with a hypothesis that the yeast fungi and the yeast farming ants have co-evolved, and that yeast phase growth is a genetic adaptation for life with ants. In human cultivars, we would call that domestication. Well, a clear domestication event in ant agriculture occurred in the transition from lower to higher agriculture, from the yellow to the blue boxes. Higher agriculture is practiced by the highly eusocial, ecologically dominant leafcutter ants, but it's also practiced by 5 genera of non-leaf cutting higher fungus farming ants. And if domestication is genetic change in a cultivar that increases its fitness in the agricultural symbiosis, then there are a number of traits that are clearly derived and that are shared by all higher fungal cultivars. This includes gongolidia. These are nutritious swollen hyphal tips that are preferentially harvested and eaten by the ants. You can just make out clusters, little clumps of gongolidia on the surface of this higher fungus garden. In addition to gongolidia, higher fungi are also polyploid, unlike lower fungi, unlike most fungi, which are usually diploid. Instead of 2 nuclei per cell, which is typical for fungi, these have more than 2 nuclei per cell. They also have increased expression of plant digesting and detoxifying enzymes, many of which are concentrated in the gongolidia. And maybe most compelling, although higher antine fungi are able to produce mushrooms and may or may not be able to reproduce sexually, we still don't know. As far as we know, they're unable to live without the ants. So in higher agriculture, both ants and fungi are obligate symbionts. They cannot live without each other. Perhaps my highest research priority is tracking down rare fungus farming ant species that have a high likelihood of providing critical new data about the evolution of ant agriculture. Because even as vast tracts of habitat are being destroyed forever, 43% of fungus farming ant species are known only from their original collections, and that means their fungal cultivars remain unknown, as do pretty much all other aspects of their biology. My first example of the value of fieldwork is the genus Mycetogroicus, the ant genus Mycetogroicus, which was only described in 2001, and it contains something like 4 or 5 species. In 2008, we knew that Mycetogroicus was the sister group to the higher fungus-farming ants, but we didn't know what fungi they cultivated, so we didn't know whether Mycetogroicus species cultivated lower or higher fungi. We didn't know if they belonged in the yellow group or the blue group. So in 2008, a group of us went to Uberlândia in Minas Gerais, Brazil, where we knew one of these species, Mycetogroicus ceridensis, occurred. We located nests and we started to dig, and we were really shocked to find that garden chambers of this species are 3.5 meters deep. That's pretty deep for, you know, on average, I'd say your average fungus farming ant garden chambers maybe a meter deep, sometimes less than a meter, sometimes more than a meter, but this was truly exceptional. So in 2012, when I needed to collect more specimens of Mycetogroicus for a genomic study, I decided that we would go after a different species, one that had been collected on the banks of the Araguaia River, which I figured would have much shallower nests because it's on the banks of a river. But I was wrong about that. Their garden chambers are also over 3 meters deep, and they're underwater in the rainy season. That's a biological mystery remaining to be explored. But this field work allowed us to identify the fungi grown by both species and determine that Mycetogroicus is a lower fungus farmer. This in turn allowed us to identify on the ant phylogeny, and now more recently on the fungal phylogeny, the precise branches on which higher agriculture arose. This will guide future genomic work to identify the genetic change that took place in ants and fungi during the transition to higher agriculture. Paramycetophylax brucki is a morphologically hard-to-place fungus-farming ant species that was put into its own monotypic genus by frustrated taxonomists. And what I mean by monotypic genus is it's the only ant in that genus, so they didn't have to figure out what it was related to anymore. It occurs in very marginal xeric habitats in the Argentinian dry Chaco. I had previously looked for it there without success. During the past 4 years, I've had the good fortune to team up with Argentinian myrmecologist Priscilla Hanisch, and she ultimately was able to locate nests of this enigmatic species, which she found consistently occurred under trees known locally as Algarrobo dulce. Priscilla became the first person ever to excavate nests of Paramycetophylax brucki. She collected ants and fungi and recorded biological information about this previously unstudied fungus-farming ant species. And when we added Paramycetophylax brucki to our UCE phylogenomic ant phylogeny, we got an unexpected surprise. It is the sister species to the yeast-farming ants, having diverged from them around 36 million years ago. That was published last year. And when we added the cultivar of Paramycetophylax brucki to our phylogenomic fungal cultivar phylogeny, we got an even bigger surprise. The fungus cultivated by Paramycetophylax brucki is the sister group to the yeast cultivars. So the ants are the Paramycetophylax brucki is the sister to the yeast-farming ants. The fungus that it grows is a sister to the yeast fungi. What this means is, if we want to explore the genetic change responsible for the origin of yeast agriculture, our search has been narrowed to 2 branches, one in the ant tree and one in the fungal tree. Aprostigma megacephala. This ant was first described in 1999 based on just 4 specimens from Peru and Colombia. We searched for it in Peru without success. Then, in 2008, colleagues told us about a specimen that had been collected in Carajás in the state of Pará in Brazil. So this was the sort of circum-Amazonian distribution of Aprostigma megacephala that was known at that time, and this was the new specimen that had the new record. My graduate student at the time, Jeffrey Sosa Calvo, who is here tonight and is now a postdoc with me, went to Carajás and he located a population of Aprostigma megacephala. Here's a picture that Jeffrey took of Aprostigma megacephala on its fungus garden. When we added Aprostigma megacephala to our phylogeny, this was the position that it occupied. And when we obtained DNA sequences for its fungal cultivar, we got what I think I could fairly say is one of the biggest surprises of our careers. Aprostigma megacephala is the only lower fungus farming ant known to cultivate a higher fungal cultivar. It actually grows the leaf cutter cultivar Leucoagaricus gongylophoros. So that harkens back to earlier in the talk when in 1890, I believe it was, Alfred Mueller named that species. Since then, in 2017, we returned, we collected in Mato Grosso, and we were able to make 2 additional, multiple colonies in 2 different locations of Aprostigma megacephala. And I can tell you that in all cases, it grows the higher fungal cultivar Leucoagaricus gongylophoros. So I believe that if we understood why Aprostigma megacephala is the only lower fungus farming ant known to cultivate a higher fungal cultivar, we might get some insight into the biological mechanisms that otherwise constrain the 5 agricultural systems. There must be some kind of physiological, nutritional, or sensory barriers that constrain these systems. So I hope that these examples demonstrate the importance of seeking out rare and poorly understood fungus farming ant species and understanding their biology, particularly what fungi they cultivate. Having reliable dated phylogenies for the ants and their fungal cultivars allows us to explore historical questions about correlations between major evolutionary transitions in ant agriculture and major events in Earth history. The origin of human agriculture was correlated with climate change. It arose all over the planet only after the end of the Younger Dryas, near glacial period 12,000 years ago. So what about the origin of ant agriculture? Our ant and fungal chronograms indicate that both lower fungus farming ants and the lower fungal cultivars in the yellow boxes here originated 66 million years ago, coincident with the end of Cretaceous mass extinction event. That's when a bolide collided with the Earth and caused firestorms for days to weeks and shut down photosynthesis for several or more months, or possibly, as recent news articles indicate, possibly even for a few years, during which the fossil record indicates that fungi proliferated and global mass extinctions occurred. As pointed out by the ecologist Dan Jansen, the types of animals most likely to survive such conditions were small, semi-subterranean detritivores or their predators, including, among the former, the detritivores, invertebrates specialized on locating and consuming decaying organic matter or scattered dormant seeds and insects. Under such conditions, fungus-farming ants and leucocypriniaceous fungi would have constituted a formidable mutualism in which the ants located organic materials and the fungi digested them, sharing the nutrition with the ants. This allowed both partners to persist through a period of mass extinctions. Interestingly, the sister group of the fungus-farming ants, which by definition arose simultaneously with them, is the subtribe Dasytina. The Dasytina are specialized predators. Some of the earliest diverging lineages in the Dasytina look like this, with these really amazing trap jaws for capturing prey. A study in 1993 concluded that it's very likely that the early Dasytina preyed on collembola, which are detritivores. So this explains how they might have survived the end of Cretaceous extinction event along with the fungus-farming ants. Interestingly, even though they arose at the same time around 66 million years ago, the two sister groups have different global distributions. The Dasytine predators on all these, I don't know if you can tell, but they're purple branches, managed to radiate across Antarctica, which was much more temperate at that time, into Australia, with one species making it up into Africa. In contrast, the fungus-farming ants, which I've represented just with this red branch here, have always been confined to the New World. I think the explanation is that fungus-farming ant distributions are limited by the cold sensitivity of their fungi. Even today, very few fungus-farming ants make it up into the northern or southern temperate regions. So whereas the Dasytines were able to adapt to the then temperate conditions in Antarctica and spread eastward, the fungus-farming ants were not, because they were constrained by their symbiotic association. So symbiotic associations can open up new ecological opportunities for you, but they can also close off others. The transition from lower to higher agriculture, which clearly involved genetic changes in the fungus, occurred around 30 million years ago on both the fungal and ant phylogenies, which agree amazingly well about this. Using the UCE ant phylogeny in 2017, we reconstructed this the biogeography of higher fungus-farming ants, and we found, not at all surprisingly, that they originated in South America. We also reconstructed ancestral habitats on the phylogeny and found, somewhat more surprisingly, that the ancestral higher fungus-farming ant lived in a seasonally dry habitat rather than in a wet forest, which had always been assumed. Significantly, the origin of higher ant agriculture, both the fungi and the ants, occurred after the terminal Eocene event around 35 million years ago. The terminal Eocene event set off a period of global cooling that resulted in the expansion of drier habitats and grasslands throughout the world. In South America, this resulted in the fragmentation of what was previously continuous wet forest. Humans have exerted artificial selection on our domesticates by, among other things, isolating them from their free-living gene pools, including by moving them away from their places of origin. This repeated pattern in human agriculture led us to speculate that the domestication of higher fungi likely occurred in a dry habitat in which the fungal cultivar was, unlike its ancestor, no longer in sympatric genetic contact with an extended population of wet forest-dwelling, free-living fungi. essentially dependent on its ant farmers. It was subjected to the ant equivalent of artificial selection, causing it to evolve into the ancestor of the obligate higher fungal cultivars. Last year, as Larry mentioned, we published this book, The Convergent Evolution of Agriculture in Humans and Insects. As I mentioned, human agriculture arose independently 20 times or more. Comparative study of these multiple examples of human agriculture, coupled with new methods like archaeogenomics and historical population genetics, have created a renaissance in the study of human agricultural evolution. The thesis of our book is that first, whether human or non-human, agriculture is a form of symbiosis, and second, that when we extend this comparative approach that's currently being applied to human agriculture to include non-human agriculture, we observe many of the same historical patterns. We think this is due not to coincidence, but to convergent evolution, because there is a limited number of ways that you can grow your own food and a limited number of evolutionary pathways that can lead to growing your own food. Here's just one example of an evolutionary sequence that is shared by both humans and fungus-farming ants. Agriculture arose in social groups of central place hunter-gatherers, in the case of humans for tens of thousands of years, and in the case of ants for millions of years. These hunter-gatherers modified their local environments and co-evolved with local pre-adapted species, often camp-following species, that thrived in the disturbed habitats created by the future farmers. This prolonged period of community coevolution was followed by incidental cultivation, in which the farmers unconsciously, and then later in humans consciously, encouraged the growth of their favored food organisms. This was followed by a commitment to cultivation, the beginning of agriculture. And this was followed by, or occurred simultaneously with, domestication, the genetic change in the cultivar that increased its fitness within the agricultural symbiosis, and in the ants, also genetic change in the ants. In humans, incomplete genetic change. We have undergone genetic change, such as lactose intolerance and things like that. In many cases, this ultimately led to the evolution of large complex societies based on agriculture. In the ants, that would be the leafcutter ants. It also led to the creation of complex agroecosystems, ecological communities consisting of hundreds or thousands of species. The agroecosystems created by adult leaf-cutting species constitute such a reliable and predictable resource that they support entire agricultural communities, including hundreds of endemic species, including like many species of beetles, of mollusks, of flies, and even 4 different species of reptiles. I'm often asked about the benefits of this research, by which the questioner usually means, how does it benefit human economics? One benefit is, at this point in time, many dozens of antibiotics have been identified by studying the fungal cultivars, the biochemistry of the fungal cultivars, the biochemistry of Escovopsis, the crop disease, biochemistry of the actinomycete bacteria. And these include antibacterials, antifungals, and even an antimalarial. A number of promising anti-cancer compounds are currently under study. Fungus-farming ants have practiced agriculture and controlled diseases with antibiotics for 66 million years, whereas humans have practiced agriculture for 12,000 years and utilized antibiotics for less than a century. Likewise, leaf-cutting ants have essentially husbanded local trees and vegetation for millions of years, stripping them of part of their vegetation, but not to the extent that they kill them. Understanding the evolutionary and ecological dynamics of ant agriculture could therefore have important and practical applications for the longer-term practice of human agriculture and the longer-term practice of human medicine, because we are currently practicing short-term strategies. Another benefit, my personal favorite, is simply the wonder and awe that we experience when we study these remarkable organisms. So I invite you to stay tuned for further wonder and awe, and I thank you kindly for your interest.
We have time for some questions, as always.
Yeah, Zemankova, member. Absolutely wonderful talk. And I love mushrooms. Okay, I don't like ants as much, especially when they're in my pants. Okay? So in human agriculture, there are poachers, right? The humans plant corn, and there are maybe not poachers, but people go and steal their corn or whatever. Or in the community gardens, they come and steal their tomatoes, etc., etc. So the question is, are there poachers in the animal world who would go and either eat the fungi that are produced or farmed by the ants, or take the advantage of seeing the fungi and eat the ants, such as the ant eaters. You know, the ant eaters look, oh, where are the mushrooms? And now I go and eat the ants.
Yes and yes. There are animals that specialize on the ants. Some of them, actually some poison dart frogs eat the ants and apparently derive some kind of basic chemicals out of them that they then use to construct their poisons. Anteaters are a good example that go after large leafcutter nests. There are some, I'm not sure there are very many arthropod arthropods that take advantage of the fungus gardens. You would think there would be. There are tons that live in the dump chambers, the waste chambers. But there are a couple of super interesting cases. There is another kind of ant in a genus called Megalomyrmex that there's a number of species, and they have a grade of strategies. I think the most primitive ones, they just go in, they raid, they decimate the garden, they eat some of the ant larvae, and then they eat the garden. So they have the ability to digest fungi. In the more sort of derived state, they move in with the ants, and they just live there. They basically are kind of husbanding their hosts and allowing them to live, allowing them to even to reproduce, but taking enough fungus so that they can do likewise. And then one of these protects. There's another kind of ant called Namptogenes that frequently raids fungus-growing ant colonies. The Megalomyrmex protects its host ants from the Namptogenes. So when Namptogene raiders come in, if you're a fungus farming ant and you're occupied by Megalomyrmex, the Megalomyrmex will ward off the Namptogenes because they have these toxins that they can express from their stings. So what do you call that? Is that a parasitism? Is that it shows you the context dependency of this symbiotic definitions that we have made.
My name is Tristan Ritland. I'm not a member. I was curious, I know that, for example, lichens are well known to be difficult to grow in captivity or in a lab. Has there been any attempt to, grow these in a more controlled environment, and has it seen success? Any cultivar?
To grow the ant cultivated fungi?
No, just the whole system, the ants and their
Oh,
fungi.
yeah, I've been doing that since I was a graduate student. Yes, and I learned from others. It isn't that hard actually I mean, it may be somewhat hard to locate some of these species. It may be somewhat difficult to excavate them, but if you can collect a whole garden or gardens, if they have multiple chambers, and if you can get the queen, get most of the workers and the queen, there are ways to keep them alive in the field. We've designed, Jeffrey's the first author on a paper of how to do this. It's basically how to culture fungus farming ant colonies. And we've designed these little plastic boxes and things that you can use. And then once you get them back to the lab, they're relatively easy to take care of. Some of them are hard because we don't know what they want. I talked about the substrates that they forage for. There's a few that we are not clear on what substrate it is they want, but for the most part, they're pretty easy to take care of. So you can culture these for years in the lab. You can isolate the fungus from the garden and grow it out in axenic culture, get it in sterile culture. You can grow up large quantities of it if that's what you want to do. We do that. You can also culture Escovopsis, and I've never tried. I think the actinomycete's harder to culture.
So I actually have a follow-up then. The cultivar the species of ant that you noted as right on the boundary of the yeast growers, has there been any attempt to see if that very closely related fungus turns into yeast if you give it to a yeast growing into a yeast growing ant?
Oh, what a great idea. No, it's very hard to work in Argentina permit-wise. I was very lucky to work with Priscilla Hanisch. She's off doing something else now. This was kind of a side project for her. Somebody needs, preferably an Argentinian, needs to get interested in this. That would be a very interesting, I would predict that they wouldn't be able to, the branch separating the fungal cultivar of Paramycetophylax brucei from the yeast cultivars is a very long branch. So it's long in terms of numbers of years, and it's long in terms of just sort of genetic change on that branch. So I would predict that something happened on that long branch that made it, yeast, created the yeast cultivars. But no one even has this fungus in culture at the moment. It's really a shame.
If you're saying that if the fungi are, if the yeast producing fungi, right, are not as, if the closest ancestor to the yeast growing fungi that is also cultivated by the ants is very far from the, if the mushrooms are far apart, but the ants are closer, you could try swapping the ants and seeing if the ants can cultivate the yeast cultivars when they revert to mushrooms, as opposed to seeing if the mushrooms revert to yeast.
Yeah.
It might be a smaller jump.
In fact, the branches in the ants and the fungi are both very long. But that would nonetheless be a very interesting experiment. And I think if you had yeast growing ant colonies in the lab and you had Paramycetophylax brucei in the lab, you could do both sets of switches, and it would be very worthwhile to do. I wish I knew someone who was interested.
We're going to go to the web first. We have some questions. It'll be the red microphone in the back there.
Thank you. The first question is from Carl Merrill. He is streaming from Bethesda, Maryland, and he is a member of PSW. His question is, could you comment on fungal strains that affect ant behavior, such as fungal that are associated with zombie ants? Could such behavioral changes be also associated with the agriculture?
Aha. Well, probably most of you have heard of zombie, I guess it's zombie ants or zombie fungi, but anyway, there's these kinds of fungi that infect ants and other insects too. There's quite a few species of these fungi. Many of them have very specific hosts. They infect, in the case of ants, they infect ants. They alter the ant's behavior. They cause ants to do things that ants would do in some other context, but not in this context normally. Namely, climb up to the top of some plant, clamp their mandibles around the end of that stalk, and just stay there as the parasitic fungus grows into the ant and then sprouts, often out of the joint between the head and the body, and of course kills the ant. And why? Well, first of all, why would the fungus want to do that? Well, broadcasting your spores from a high spot is always better than broadcasting your spores from a low spot. That's why it's good for the fungus. How do they do that? Well, that's something that people are very interested in, and a number of people are studying now. Like, I mean, is there some simple biochemical explanation, maybe one or two molecules that would induce this fairly complex behavior in an ant? But the bottom line is these fungi are able to manipulate the behaviors of ants and other insects too. Could this be happening with yes, it could be. I mean, it seems unlikely, but I've worked for with mycologists since, well, and maybe for mycologists too, but with mycologists since I was a graduate student, and their take on the system is, oh yeah, there's the fungus garden down there is kind of like a brain sitting there sending out its ant minions to bring it food, And that's a perfectly legitimate way to look at it. Who's in charge? I mean, probably, neither. But so does the fungus, I mean, there are actually a few papers that claim to show that the fungi are manipulating the ants, or at least communicating with the ants and telling them like, hey, I'm sick of that substrate, I need a new substrate. There are other explanations for what's actually going on there though. But anyway, yes, it remains possible that the fungi are directly affecting the behavior of the ants. I mean, there's no evidence for that, but it could be.
Resistance is futile. Another question from the web, and then we'll go to the blue microphone.
The second question is from Jim's Talons. He is streaming from Boynton Beach, Florida. He is not a member. The question is, human antibiotics lose effectiveness as bacteria change through evolution. Does this not happen with ants?
Could you tell me the first part again?
Sure. The first part is human antibiotics lose effectiveness as bacteria change through evolution.
Yeah, that's a great question. And we've wondered about, ants, you might say ants have been using the same antibiotics for 66 million years. Is that plausible? I imagine the same dynamics apply to antibiotics as do human antibiotics, that there's evolution going on. So what we think is happening is it's an arms race. That one strategy is the ants do something. It's an improved way of controlling their parasite, and then the parasite adapts to that, and it's an ongoing, never-ending arms race. And that's our most likely explanation, but given that it's a fairly complicated context, there are other possibilities. What we do know is that there are multiple species of Escabopsis. So it isn't like a single species of Escabopsis is engaged in a arms race with multiple species of ants. And then there's the garden cultivars. It could be much more correlated with what cultivar the Escovopsis is infecting than with the ant that's growing the cultivar. But the best we can come up with is that it's some kind of never-ending arms race.
Thank you so much for your important research and this fantastic presentation. It's hard to follow zombie fungi. My question is about benefits. So with the decline of certain arboreal ecosystems, very specific tree species that we know also have similar symbiotic relationships with fungi in their root systems, in places where there's very urgent decline, is there any application for your research where you might be able to bring in some rescue ants and their cultivating skills to help bring back the soil for these tree species that are in decline and are under attack from various forces, climate change, human encroachment, etc.?
Well, a number of things occur to me. If it's in an area of the world where fungus farming ants don't occur, then it's probably like the answer would be no, there's probably nothing my research could do to help. If it's in an area where fungus farming ants do occur, and there's lots of trees in the Neotropics, then maybe, but it probably wouldn't be because the ants have any kind of association with the fungi you're talking about, the symbiotic root fungi. But it's remotely possible that it could, leaf cutting ants and fungus farming ants in general are huge, they turn over the soil where they occur in the tropics much more than any other organism, like earthworms up here, maybe. Down there, it's fungus farming ants, and I've been working in the field for 35 years or more now, and when we dig up fungus farming gardens, the gardens of fungus farming ants, as you saw. And as we're digging up one, we often break into another one. And I started thinking like, oh man, if I had X-ray vision here and I could just look all across here, there would be thousands of gardens down there. So these ants are removing soil. Often it's, well, it depends on where you are, but I was going to say it's clay soil, often in the rainforest. And then they're taking down organic matter. So in that sense, there's definitely improving the health of the soil. So in a broad sense, maybe, but in the narrower sense of doing something for those mycorrhizal fungi, probably not.
Red microphone.
Timothy Thomas, I am a member of the society. I note that the ants seem quite intelligent. When I look at the large hive and the things I've read, the colonies are clearly quite able of acting intelligently when they're threatened and when they change and so forth. And you asserted clearly that the ants were not conscious. And I'm wondering how you know that.
Oh, I guess you're referring to the part where I was talking about these various stages that led to agriculture and the ants not consciously choosing to cultivate. And I certainly didn't mean to imply. What I meant to imply by that is that if you're explaining the evolution of behavior, It's pretty simple natural selection, like a colony arises that does things a little differently, and that little bit of difference gives them some kind of advantage. They leave more offspring in the next generation. That behavior survives, the other one doesn't. In that way, it's unconscious. But do individual ants have consciousness? As someone who's put a lot of ants into alcohol, that's a very scary idea, but I would have to say it's possible. There's a book out called The Mind of a Bee. I have not read it, but apparently that researcher broaches this subject and feels that, I mean, it's also true that individual ants have personalities. Colonies, they have colony level personalities. It's become this thing in ant behavior now, or maybe in general behavioral ecology. The term personality has become a sort of an official defined term now. So I would not want to say that an insect, they have very complicated brains.
I was actually thinking about the colony.
Oh, the colony. Oh,
the colony, and where each of the ants is a neuron in this brain.
Oh, gosh.
And they're communicating and passing messages, and out of the whole mass of them, as out of the whole mass of our brain, something emerges.
Oh, well, I had never quite thought about it like that, but I will tell you that emergent colony level behaviors could only happen when you have lots and lots of little units communicating with one another chemically, some tactile communication, some context-dependent communication. You start a task, an ant comes along, sees an unfinished task and joins in. There's so many things like that going on at the colony level so individual insects, there's lots of those out there, non-social insects, that could never, it's possible they couldn't do agriculture, actually. If you look at who's doing agriculture, they all are either social or family groups, but whether that, so for sure, colony level behavior responds to stimuli. Oh yeah, I mean, if you find a leafcutter nest and you start digging into it,
They react.
yes, the big soldiers all converge on you, and that's because of alarm pheromone propagating downward and then back upward again. But whether that means that at the level of the colony there's something akin to consciousness, that would be amazing. There was a 19th century guy, is it Maurice Maeterlinck? He wrote a book about, I think about this, the mind of, it was something about a bee colony having an emergent, like consciousness, yeah.
My name is Cheryl Lohman. I'm a member. We talked about awe and wonder and maybe a fun question you could entertain. Also segueing on the topic of consciousness. You talked about the pharmaceutical industry being interested in antimalarials, antifungals, antibacterials. They're also interested recently in new types of mushrooms. Could you please comment on any psychedelic ants you've run into?
Yeah, we were just talking about this in the lab recently. Somebody asked me, had I ever eaten the ant fungi, and I had to admit I haven't, and it's not because I wouldn't want to. It's just never happened. I doubt, however, that any of the ant-cultivated fungi are psychedelic, but, you know, it'd be a much better story if they were, because you could say like psychedelics were involved in the origin of this like elaborate behavior. But no, let's see, Yeah, I don't know about psychedelics. There are some insects that produce compounds that ants imbibe and become sort of more narcoticized rather than psychedelicized. So there's a what do you call these things? Ambush bug that has a gland in the center of its chest, and ants come up and feed on that gland, become kind of stupefied, and then the bug eats them. There's a few other examples like that. Oh yeah, there's these lycaenid caterpillars that produce these substances that maybe they're just sweet or tasty, but it's possible that they're also mind-altering in a way. As for fungi, the reason, I mean, one of the proposed reasons for why some fungi express psychedelic drugs is that they're insecticidal. They're insecticidal. They repel insects. But I'm not aware of any ants that eat fungi and then trip out. I can't think of any. Is that what you were getting at? Yeah. Okay.
You've never seen a fiddler ant?
What?
Fiddler, or what is it? There's some insect that's called the fiddlers, and I can't remember what it is. Fiddler. Never mind. Red microphone.
Fiddler.
Hello, this is David Rosen. I'm a lifetime member. What is the defining difference between high agriculture and low agriculture? High and low. What's the difference?
Yeah. One of these days, we're probably going to have to dispense with that terminology. It's just that it's very baked into tradition. There was actually a paper just like came out about a month ago decrying the use of higher and lower when applied to termites. So that's pretty close to home there. But what we mean by it is, if you look at the phylogeny of all fungus farming ants, there is a derived group within that phylogeny that practices higher agriculture, that grows the higher cultivars, and we call those the higher fungus farming ants. That leaves behind all the other ants, which are not a monophyletic group, because it arises from within them. That group, if you split off the yeast fungi and the coral fungi, that group is the lower fungus farming ants. And then if you look over at the fungal phylogeny, they're growing one monophyletic group of fungi, the clade 2 lower fungi, and they're growing the clade 1 lower fungi, which are not monophyletic. They too are paraphyletic and give rise to two derived groups, the yeast fungi and the higher fungi. So I don't know if that makes sense, but that's what we're trying. We're just trying to make a distinction. And in some ways, it's like the higher fungus farming ants and everybody else, except not the yeast growers and not the coral fungus growers. So,
Did you say that some of the yeast, when transplanted to a different ant colony, they grow filaments, like they become mycelium?
I did, but not when they go into different colonies. So if you turn over a log or something and you find a yeast growing colony, you'll see all those little nodules, but then if you look at the larvae, they're covered with mycelium. So the same fungus, that's all one organism, that fungus. It's expressing itself as mycelium and as yeast in the same colony. And then in addition to that, I mentioned that we have found free-living mushrooms that when we DNA sequence them, they fell within the yeast clade of mushrooms. Those are clearly growing as mycelium in the leaf litter. They're not growing as in the yeast morph.
So it's epigenetic.
Could you please hold the microphone up so we can hear you?
Is it like epigenetic, all these differences, the same?
Yeah. Somebody needs to figure that out. My current model is this, that the yeast fungi, the ancestor of the yeast fungi was a typical lower fungus. It genetically it co-evolved, and its genetic architecture changed so that now it can express the yeast morph. And here's the way I think of it. It detects, like, oh, I'm now being cultivated by ants. I'm going to express the yeast morph. Or, oh, I'm out in the leaf litter now. I'm going to become a mycelium. And that's entirely possible. And most of our domesticates, correct me if I'm wrong, don't really have that. They don't sort of have, in general, they don't have that. They can't. So when we used to think, oh, well, we found these mushrooms growing in the wild. That must mean that the yeast fungi are facultative symbionts. They're not. I think that's a simplistic way of looking at it.
My name is Al Ehrlich, and I am a member. And I'm having trouble figuring out what exactly is meant by cultivate in your jargon. I'm not seeing the distinction between cultivate and coexist or symbiotic relationships. What do the ants do that can be considered cultivation? I mean, cultivation, I thought I knew the definition of in human farming terms, but the ants don't prepare something or move something at an appropriate point of development. In what sense are they cultivating it? And why is that term used? Or maybe I don't know what cultivation is. I don't.
Well, I think the ants do a lot to prepare a substrate on which they plant fungi, weed out foreign fungi and bacteria so that the fungus can flourish and not have competitors. They propagate the fungus vertically from mother to daughter nests. They do pretty much everything that I would think would be included in a definition of human cultivation. One thing that I think is different, and it's worth pointing out, is that they go out and, hunter-gathering ants, that's the sort of typical state in ants. They go around, forage, bring food back to the nest, and eat it and feed it to their young. These ants have not, because they've become agriculturalists, gotten rid of the need to forage. They still have to go out and forage, but now it's for substrate for their fungus garden. But then again, when you think about it, the extent to which we prepare soils now for our crops, our huge industrial monoculture crops, it's almost like that. I mean, we don't go forage for soil. Well, maybe we even do, but we bring in chemicals, we bring in all kinds of stuff to prepare the substrate on which we then plant our
I must have missed the part where you said they do these various things which are like cultivating the soil.
They're definitely not just co-occurring passively together. The fungus would not be there if the ants didn't have all of these behaviors for promoting its growth. Yeah.
Thank you.
So my two last questions, and then we'll wrap up. Does Smithsonian still have a leafcutter ant exhibit? They used to.
They used to have a fabulous one at the zoo in what was then called the invertebrate house. That's gone. But we do have one in the insect zoo in the Natural History Museum.
And which ants are in that one, and what fungus do they grow?
It's a leafcutter ant colony. It's the genus Atta. I don't know the species, and it's Leucoagaricus gongolophorus, the Alfred Mueller's species. Yeah.
It has a long tube, and you can see the ants going.
Yeah, let's see. I'm not sure it has a long tube. It has like two big exhibit cases. One of them has usually, I haven't been there in a while, it has a plant that the ants are actively cutting. And then the other one has these sort of chambers, man-made cutaway chambers where the ants are growing gardens. I don't know about a tube, but
Yeah, they used to have a tube. You could see the ants.
was that in the zoo or was
I
it?
think it was in the museum.
Oh,
That's my recollection. It was like a mold, and that was like 30 years ago.
yeah, well.
The other question is, and probably the answer is no, but do you see any evidence of selection by the ants for better fungal species or?
For better fungal species?
Yeah.
Yeah, we've thought about this for a long time. And there's certainly the opportunity for that. If you think about it, every generation, the fungus goes through a bottleneck. So even though the fungus is being clonally propagated, the ants are not allowing it to sexually, I didn't talk about this, but in the lower fungi, there's definitely sexual reproduction going on. That's got to be taking place when they're not associated with ants. So the ants are preventing any kind of sexual reproduction, but they're clonally propagating the fungus. And then they clonally propagate it across generations. And when they do that, it's a very small amount of fungus taken in the mouth. And at that point, artificial selection could be taking place. There's no reason to think that's happening. We don't know that. I mean, I would be really shocked if the daughter queen didn't go to the most healthy, luxurious, vibrant part of the garden and take her nucleus from there. But other than that, and then you got to think about the mutation rate. Like in a clonally propagated thing like that, there's lots of cells, but how many mutations occur, say, per colony life cycle or something like that, and then how many of those mutations just wink out and are not propagated? So,
Well, it's a subsidiary question. It's a footnote, but I've always wondered about mushrooms and fungi that produce all of these very complex organic substances, which when you test them in the laboratory turn out to be the most carcinogenic and mutagenic things known to man. And how do they maintain the integrity of their own genome? And how much do these substances contribute to their mutation rate? So I don't know.
yeah, I don't know anything about that.
It's an interesting Bruce Ames story, right? Because he invented the mutagenesis test using chopped liver to metabolize the substance he was testing. And one day he asked, he's testing all these chemicals, right? Because they're all worried about chemicals. So one day he asked himself, well, let me do a control experiment and see, what are the normal things? So he went to the supermarket and he bought a bunch of vegetables, including mushrooms. And he told me the most carcinogenic thing he ever tested was a mushroom.
Oh my gosh.
So,
Was it your typical button
it's just
grocery store mushroom? Like,
I didn't I think in those days that was probably all you could buy. It was, pre-gourmet mushrooms. So anyway, thank you so much for a really, really interesting talk.
Thank you.
Now, before you go, we do have a few small gifts. You don't have to declare them because they have no monetary value, but they have a lot of other kinds of value. So they are the rosette of the Philosophical Society, which you can wear like I'm wearing it. And it's interesting because the founder of the Philosophical Society was Joseph Henry, and Joseph Henry was well known for studying induction, and the symbol for induction is a coil. And the sides of the rosette are the three bars of the coil. The other founder was Joseph Henry, as you probably know, was his first secretary of the Smithsonian.
Yeah?
Yeah, so
Wow.
The two institutions have something in common, Joseph Henry. The other two things are a framed copy of the announcement of your talk and a signed copy of the volume 1 of the Bulletin of the Philosophical Society of Washington, which we now fondly know as PSW Science. And that is interesting because it says who the founders were and why they called it the Philosophical Society and why they founded it. But it has a very nice description of how we should conduct scientific meetings and discourse, which I think is very germane to today. So again, thank you so much for this talk. It was really great to have you here. So before you go, we have just a few closing announcements. The 2485th meeting will be held on November 17th. The speaker will be Bob Truog of Harvard, and he will be speaking on defining death, science, medicine, belief, and law. The subject involves a complex mix of competing viewpoints and is a microcosm of how these differences are being resolved. The 2486th meeting will be on December 1. The speaker will be Missy Cummings. She'll be speaking on the good, the bad, and the ugly of self-driving cars. Is better than humans enough? Misty is at George Mason University, where she is director of the Mason Autonomy and Robotics Center, professor of mechanical engineering, of computer and electrical engineering, and of computer science. Interestingly, prior to her academic career, she was an officer in the Navy and was one of the first women Navy fighter pilots. I think in her day, she flew Tomcats. She probably flew them right over where I was eating lunch at the Scripps Research Institute, right next to Torrey Pines Park, adjacent to the ocean. Anyway, please check the PSW website often for up-to-date information on meetings and lectures. Let's thank our volunteers who worked hard tonight to do all the things that need to be done to have these meetings and lectures. And if any of you out there would like to help with the PSW activities, please see me. There's a lot of things to be done, and we're looking for volunteers. With that, I will entertain a motion to adjourn the meeting to the social hour. Have a second? All in favor? All opposed? Meeting is adjourned. Thank you all for coming.